and welcome to my new session that is on hybrid seed production, principles and practices. Myself, Dr. Narkade Gopal Vasudev, working as Assistant Professor, Department of Genetics and Plant Breeding, School of Agriculture, SR University in Warangal. So today, in this session, we will see the principles and practices for hybrid seed production. So first, we will see what is hybrid. So the hybrid is the progeny. Hybrid is the progeny of a cross between genetically different plant. In other words, you can say like hybrids are the resultant product or hybrids are produced by crossing two or more genetically dissimilar parents. That whatever the output will come, that output product is called as hybrid. And what are the hybrid varieties? The F1 populations that are used for commercial cultivation, those are hybrid varieties. Hybrids are the major kind of varieties that are ruling the seed industry at present. Right? So instead of varieties, hybrids are ruling the seed industry. So they are categorized as high value, low volume crops, and are in great demand due to 100% seed replacement ratio. So what is this 100% seed replacement ratio? That means farmers can't use the self-saved seeds from hybrids, unlike the varieties or open pollinated varieties. Every season or every year, farmer needs to replace the seed. So that's why 100% seed replacement ratio is there. The F2 generation results in segregation of characters, producing ununiform crop growth. That's why we'll call F2 generation of crop as the most segregating generation in plant life cycle. Right? What are the main features of hybrids? So as now I told, hybrids are ruling the seed industry. So what are the main features? So first, most important, that is productivity. So hybrid varieties are vigorous and highly productive because in hybrids, heterosis is fully exploited. So what is heterosis? Heterosis means superiority of that particular hybrid for yield and yield contributing characters over the parents. If your hybrid is superior in terms of yield and yield contributing characters over both the parents, that means your hybrid is vigorous and highly productive. Then second point, it is genetic constitution. So all the plants of hybrid variety are genetically similar, right? They all are heterozygous in nature. They are phenotypically uniform and more attractive due to homogeneous nature, right? Then third main feature is adaptability. So hybrids, they are having wider adaptability to the environment change. Whatever the fluctuating climate is there, hybrids will possess wider adaptability when we will compare with inbreds and pure lines. So we can develop hybrids in both self and cross-pollinated species. These hybrids, they are generally more tolerant to biotic and abiotic stresses when we'll compare the performance of inbred or pure varieties, that is pure lines. Then our next point is commercial cultivated hybrids. So generally these commercial cultivated hybrids are of two types. First, intraspecific hybrids. So what is intraspecific? Like when a hybrid between genetically different genotypes of same species. A hybrid between genetically different genotypes of same species. It is known as intraspecific hybrid. This intraspecific hybrid is also called as intervarietal hybrid. So this type of hybrids are commonly used for commercial cultivation. And this type of hybrids are always fertile. So that's why they are commonly used for commercial cultivation. The second type of hybrid, that is interspecific hybrid. What is interspecific hybrid? The F1 progeny between two different species of same genus. The F1 progeny between two different species of same genus is known as interspecific hybrid. This interspecific hybrid also called as intrageneric hybrid. So this interspecific or intrageneric hybrid, they are rarely used for commercial cultivation because, as I told you, intraspecific hybrids are always fertile. But these interspecific hybrids, they are fertile only in few cases. That is, for example, Lemquit, that is a hybrid variety from the crossing of key lime and Comaquit. Let's see some types of hybrids. So before that, we'll see what is meant by pure line. What is pure line? This pure line is the progeny of single self-fertilized homozygous plant. That's why the name itself it indicates pure line. It is the progeny of single self-fertilized homozygous plant. Next, what is meant by inbred line? So, inbred line is a near homozygous line obtained by continuous inbreeding in cross-pollinated crop, followed by selection. Whenever we will do continuous inbreeding in cross-pollinated crop, and we will select some desirable lines. Those lines are called as inbred lines. So those lines are also almost homozygous. Then we'll move towards types of hybrids. There are three types: single cross, double cross, three-way cross. So what is single cross? The single cross hybrid progeny is a progeny developed from a cross between two inbreds or two varieties. Whenever we cross two inbreeds or two varieties, the resultant F1, it is called as single cross hybrid. Next, what is meant by double cross hybrid? The hybrid progeny from a cross between two single crosses. Whenever we'll cross the two single crosses among themselves, two different single crosses, that means involvement of four parents. Whatever the resultant progeny we will get, that is called as double cross hybrid. Right? Then next, what is the three-way cross? Three-way cross means a hybrid. Three-way cross hybrid means that is a hybrid progeny between single cross and inbred line. That means in single cross, involvement of two parents are there. In double cross, four parents involvement is there. And in three-way cross, single cross means two parents plus inbred line. As the name itself indicates, three-way cross hybrid. That means involvement of three parents. So these are the types of hybrids. Then next is top cross. Now, what is meant by top cross? Top cross, it is a cross between an inbred line and an open pollinated variety. The purpose of this top cross is the estimation of general combining ability of inbred line. Right? When the cross is made to assess the combining ability, it is known as test cross. A test cross may be made with an inbred for specific combining ability, hybrid, synthetic or open pollinated variety for GCA. The common parent used in the test cross is known as tester. And the progeny derived from these crosses, they are called as Just cross progeny. Next type, that is polycross. This polycross, it is the progeny of line produced through random pollination by number of selected lines. That means whatever the resultant progeny we will get through random pollination by number of selected lines. So that progeny is called as polycross progeny. Next type of cross, that is varietal cross. As name itself indicates, when two open pollinated varieties are mated, it is known as varietal or population cross. Right? So now we have seen some different types of hybrids. Now we'll come towards our main title, that is hybrid seed production. Now here we will see what are the requisites. What are the requisites of hybrid seed production? First, breeders. What are the breeders' responsibilities? So in breeders' responsibilities, first development of inbred line. Next, identification of specific parental lines. And third, develop system for pollination control. So these three are the major responsibilities of the breeder. Right. So in second point, in requisites, that is major problems for breeders and producers. What are the major problems? So foremost and most important problem, maintenance of purity of parental lines. Then next, emasculation, that is separation of male and female reproductive organs. And third, pollination system. These three are the major problems breeders and seed producers will face. What are the characteristics? Now, generally for hybrid seed production, we'll use male and female parent, right? So, what should be the characteristics of the parental lines? Right. So, for female parent, the female parent should be high yielding. Good seed characteristics. What if seed characteristics means? Seed should be shiny, having luster, small, or if we are targeting bold seed. So it is based on our objective, lodging resistance. Then what are the characteristics of male parent? Good pollen production, long shedding period, plant height, and if we are dealing with male sterility, then fertility restoration. So these are the some major criteria or characteristics of the parental lines. Then the basic principles of hybrid seed production. There are two basic principles, like first, you need to avoid the self-pollination in seed or female parent. You need to avoid the self-pollination. And the second, you need to ensure crossing female parent with desirable pollen parent. No blind crossing. You need to cross the female parent with the desirable pollen parent only. Right? Now, development of hybrid varieties. So this development of hybrid varieties differ from species to species. This breeding for hybrids generally involves three steps. First, development of inbred. Next, evaluation of those inbred lines. And third, production of hybrid seed, commercial utilization of the crosses for seed production. So these are the three main steps involving hybrid breeding. Now first, one by one we will see. First, development of inbred. So it is important step in production of hybrids. There are two methods of developing inbred lines. That is, first one by selfing of heterozygous population. Just we have discussed in earlier slides. That is inbreeding in cross pollination, inbreeding in cross pollinated crops. Then second, by doubling of the haploids. So these inbreed lines are developed by continuous self-fertilization of cross-pollinated species. This inbreeding of an open-pollinated variety leads to many deficiencies. So if you will do inbreeding in open-pollinated varieties, ultimately it will leads to loss of vigor, reduced plant height, and due to continuous inbreeding, these plants will become susceptible to lodging, insect pest resistant, insect pest attack, and many other undesirable characters. So after each selfing, you need to select the desirable plants and promote them for self-pollination. So in this way, selfing, you need to continue this selfing for 6 to 7 generations to get the superior homozygous inbreed. Okay. So an inbreed line you can maintain by selfing. These inbreeds can also be developed from haploids by doubling the chromosome number through colchicine. So you can double the chromosome number of crop by using the chemical called colchicine. So with this use of colchicine treatment, it is the shortcut method for developing inbreeds. The original self plant is generally referred as S0 and the first self progeny referred as S1, second S2 and so on. So the techniques of inbreeding requires careful attention to prevent the natural crossing. The inbreed lines are identified by numbers, letters, or you can put combination of both. Right. So for development of inbreeds, either go for selfing or go for colchicine treatment for doubling of the chromosomes. Now, second step, evaluation of the inbreed lines. So after an inbreed line is developed, it is crossed with other inbreeds and its productiveness in single and double cross combinations is evaluated. The ability of inbreed to transmit the desirable performance to its hybrid progeny. That is called as combining ability. What is combining ability? The ability of that particular inbreed to inherit or to transmit the desirable characteristics into its hybrid progeny. That is called as combining ability. So generally now, I think you may have aware like combining ability, it is of two types, general combining ability where we will evaluate parents and specific combining ability we will evaluate to test the hybrids. So these inbreeds are evaluated in following ways like first phenotypic evaluation. So it is based on phenotypic performance of inbreeds. So this phenotypic evaluation is the effective for characters which are highly heritable. That means high general combining ability. The poorly performing inbreeds you need to reject. Right. The performance of inbreeds is tested in replicated yield trials and inbreeds showing poor performance you need to discard. The second evaluation method, that is top cross test. As I told, inbreeds which are selected after phenotypic evaluation, you need to cross them to a tester with wide genetic base. So example for tester with wide genetic base means either you can use open pollinated variety, you can use synthetic variety or double cross. The performance of this top cross progeny is evaluated in replicated yield trials, preferably over location and year. So that means you need to go for multi-location, multi-year, multi-location trial. Right. That means you need to check the stability of those inbreeds. Based on this top cross test, about 50% of the inbreed are eliminated. This reduces the number of inbreeds to manageable size for next step. Sorry. Top cross performance provides the reliable estimates of GC. Then next, single cross evaluation. So outstanding single cross combinations can be identified only by testing the performance of single cross. The remaining inbreed lines after top cross test are generally crossed in diallele or line into test or mating design to test for SCA, specific combining ability. A single cross plant are completely heterozygous and homogeneous. That means they are genetically similar and they are uniform. A superior single cross regains the vigor, productivity that was lost during inbreeding. As we have discussed earlier, like F1s are more vigorous and high yield and can be more vigorous and productive than original open pollinated variety. Right. The performance of single cross is evaluated in replicated yield trial. Again, you need to evaluate the single cross multi-year, multi-location to check the stability. Outstanding single cross identified and may be released as a hybrid where production of single cross is commercially feasible. Then fourth method, that is prediction of performance of double cross hybrids. So in a double cross hybrid, 4 inbred parents are involved. Theoretically, the potential of the double cross will be the function of breeding value of these 4 parental inbreds. So as I told you, in double cross means cross between 2 single crosses. That means involvement of 4 parental inbreds. Therefore, based on the procedure of testing, the breeding value of inbreds, performance of double cross hybrids can be predicted through any of the 4 methods given by Jenkins in 1934. So, which are those 4 methods? First, top cross testing. Second, mean of 4 non-parental single crosses. Right. Then third, average yield performance of all possible 6 crosses. Then last method, average progeny performance of each inbreed. So these are the 4 different methods by using you can predict the performance of double cross hybrids. So these methods given by Jenkins in the year 1934. Now the third step, that is production of hybrids. So this commercial hybrid seed production demands the crossing technique, which is easy and also economic to maintain the parental lines. So only few crossing mechanisms have been adapted for commercial hybrid seed production. So in that, first the basic mechanism, that is hand emasculation and pollination. So generally we will follow. For example, I kept some random crop names, that is tomato, chili, okra, brinjal and potato. Then second mechanism, self-incompatibility. Example, cabbage, cauliflower, muskmelon. Declining are sex expressions, that is in papaya and cucurbits. Then last, male sterility. Examples like tomato, chili, onion, carrot and rice. So these techniques are specific to crop floral biology and flowering behavior. These techniques have their own advantages and disadvantages. So based on the crop behavior and crossing technique have been adapted for production of hybrid seeds commercially. So among these different techniques, self-incompatibility and sex expression have significance, particularly in vegetable and flower hybrid seed production. So we will see one by one. So first mechanism for hybrid, that is hand emasculation and pollination. So this is manual method, as name indicates, hand emasculation. So this is the manual method wherein hermaphrodite flower. So what is this hermaphrodite flower means? That is bisexual flower. Wherein the hermaphrodite flower, androecium we need to remove. Androecium means male part. We need to remove without damaging the gynoecium, that is female part, to avoid the self-pollination in seed parent. So this emasculation is done to avoid the self-pollination in female parent. So this method is applicable in crops where flower size is handy and flower structure favors the removal of anther without damaging the stigma and ovule. So for success of this technique, generally 3 steps are involved. So first you need to do emasculation, you need to collect the pollen, and at last you need to go for pollination. So emasculation means what? Removal of anthers in flowers of female parent without damaging the gynoecium before anthesis. That is called as emasculation. Usually emasculation is done during evening hours between 3 to 5. You need to select the flowers which are going to open tomorrow for emasculation. Selection of flowers is based on morphological indications of the flower bud. Then second step, as I told, collection of pollen. The pollen has to be collected from pollen parent. Freshly opened flower can be collected and used for pollination. All flowers yet to open can be collected during evening hours and dried under 100-watt bulb to dehydrate the anthers. And fresh pollens are collected to pollinate in the next morning. Then third step, after collection of pollen, that is pollination. So hand pollination is done by hands to emasculated flower in the female parent. The emasculated flower stigma will be receptive to receive the pollens, and this stage pollination will be effective. Usually pollination is done during morning hours depending on the time of anthesis. Once pollination is done, the crossed flowers are marked either by removing one calyx in tomato or by tying the thread, for example in chili, brinjal, okra. So you need to do marking after crossing. This marking will help to harvest only cross fruits during harvesting, which helps in avoiding the self fruits and thus helps in maintaining the genetic purity of hybrid seeds. The number of fruits per plant for emasculation and pollination depends on the mother plant nutrition. The second method, that is male sterility. The inability of bisexual flower to produce the viable pollen. So this system is male sterility. So this system helps in keeping of emasculation process. So that will avoid the self-pollination in female parent, and only pollination has to be done manually. So male sterility is classified into 3 major groups, that is genetic male sterility, morphologic male sterility, and environmental genic male sterility. So next, types of male sterility based on phenotypic expression of gene. So there are 3 types. Structural male sterility. In structural male sterility, stamens are either absent, malformed, or modified into other floral parts, which leads to absence of microsporogenesis. For example, carrot and onion. Second, sporogenous male sterility, in where stamens are formed but there is an absence of pollens due to abortion of microsporogenous cells, which occurs before, during, and after meiosis. The third type, that is functional male sterility, where the pollen is viable but a barrier prevents the fertilization. For example, tomato, brinjal. Then, based on inheritance, there are different types of male sorry. So based on inheritance, there are different types of male sterility. That is genetic male sterility, cytoplasmic male sterility, cytoplasmic genetic male sterility, and chemically induced male sterility. So first we will see genetic or nuclear male sterility. We'll call it as a GMS. So this GMS having wide occurrence in plants, mostly governed by a single recessive gene. Exception is safflower, where genetic male sterility is dominant. These male sterile alleles arise spontaneously or may be artificially induced. The next type, that is cytoplasmic male sterility, determined by cytoplasm. This CMS system is the result of mutation in mitochondrial genome. Then third type, cytoplasmic genetic male sterility, CGMS system. So this nuclear gene restores the fertility in male sterile line. It is also known as nucleoplasmic male sterility. In CGMS system, we need fertility restorer, that is R gene. Then fourth type, chemically induced male sterility. You can induce male sterility by using chemicals like IAA, IBA, ethyl, etc. What are the sources of male sterility? So there are different sources like natural population. Artificially you can induce through mutagenesis. Again, genetic engineering and protoplast fusion. The male sterility system used for the development of hybrids involves 3 lines, that is 3-line breeding system. Generally all these 3 lines are present in cytoplasmic genetic male sterility, CGMS. So that is A line, male sterile, B line, maintainer line, and R line, that is restorer or a pollinator line. So basic steps in use like A line, female parent of the desired genetic background, genetically sterile. B line is called maintainer. So this is an isogenic line in the genetic background, as that of A line, and its function is in the maintenance. So A and B line, both lines are called as isogenic, differing for single cytoplasmic gene. What is R line? This is male parental line and also the best specific combiner with A line. So this is restorer line. Right. The next, self-incompatibility. This SI is the mechanism which avoids the self-fertilization through recognition of self-pollen in or on stigma of the female pistil. In a hermaphrodite flower, there is a no seed set upon self-pollination, that is called as self-incompatibility. But when pollen from other plant with different genetic content carried by wind or insects are accepted and sets the seeds, this self-incompatibility prevents self-pollination, that is inbreed, and promotes cross-pollination and creates the genetic variability. This self-incompatibility is a widespread mechanism in flowering plants that prevents the inbreeding and promotes outcrossing. Then fourth method, sex modification through hormones and chemicals. The sex expressions in plants is genetically determined and can be modified to a considerable extent by environmental and external factors such as mineral nutrition, photoperiod temperature, and phytohormones. Among these, phytohormones have been found to be most effective agents for sex modifications, and their role in regulation of expression in flowering plants has been documented. So, hormones and chemicals inducing femaleness, that is auxins, naphthalene acetic acid, ethyl, ethepone, in cytokinins like BA. Then brasinosteroids. Hormones and chemicals inducing maleness, that is GA3, AGNO3, ABA, thiopropionic acid, thalidomide, paclobutrazole. So these are some hormones and chemicals. So environmental sex modifications for seed parental multiplication. So this environment also has a greater influence on sex modification. In cucumber, high temperature and long day length favors the male flowers, high temperature extend the flowering of female flowers. So this clearly indicates that high temperature favors male and delays the female flowering. So male sex expressions of several plant species is favored by high temperature and female sex expression by low temperature. So methods of planting system in hybrid seed production. So first, block planting. Parental lines are planted in block system, which favors the hand collection of pollen and pollination system. The area under female parent is more than the area under male parent. This block planting using hand emasculation and pollination technique method of hybrid seed production. Second method, that is inter-row planting. These parents are planted in separate rows as inter-row planting based on planting ratio. These rows of female parents are more than male parent. It is used in male sterility system and self-incompatible lines with natural pollination or insect pollination. The next concept, that is planting ratio. The planting ratio depends on the pollen load required for complete the pollination and fertilization. So this planting ratio will vary from crop to crop. Border rows. These male parents are planted all along the hybrid seed production plant. This helps to avoid the cross contamination for female parents and act as an additional pollen source. Synchronization of flowering. To achieve good pollination and seed setting during hybrid seed production, there should be synchronization of flowering stage of seed and pollen period. The difference in the flowering period will lead to non-availability of sufficient pollen for pollination of seed parent, resulting in reduced seed yield. Hybrid seed production involves using genetically dissimilar parents, and hence there will be a difference in flowering period also. Hence, there is a need to synchronize the flowering period for successful hybrid seed production. Following are the methods adapted to achieve the synchronization. First, staggered sowing. The seeds of late flowering plant will be sown earlier than the seeds of early flowering parent by keeping required interval. So for example, if the male parent flowers 50 days after the sowing and female parent flowers 30 days after sowing. So you need to sow, male parents will take more time. So the difference between two parental lines, that is 20 days. Hence, late flowering male parent is sown 20 days before than female parent. So that both parents will flower at same time. Sometimes, to increase the availability of pollen, the male parent is staggered like 20 days early and 15 days early than female parent. The next, pollen storage. Pollen of early flowering male parent is collected and stored under cold storage condition with saturated CO2. Stored pollen can later be used for pollination when the late flowering female parent begins flowering. Third, adjustment in the cultural operations. Cultural practices can be used to modify the flowering time, but adjustment should not exceed 5 to 7 days. To induce the flowering in late parent, you can apply higher phosphatic fertilizer, and also you can withhold the irrigation. To delay the flowering in early parent, you can apply more nitrogenous fertilizer, and you can increase the irrigation frequency. So the nitrogen spray 0.5 to 2% can also influence the flowering behavior. Then next, most important, roguing. So roguing is the removal of off-type plant from both male and female parent to maintain the genetic purity of hybrid seed. So timely and adequate roguing is essential for ensuring the high quality seed production. The number of roguing operations depend on crop type, purity of planting seed, and stages of seed multiplication. Roguing can be carried out at different crop stages like vegetative stage, flowering stage, and maturity stage. Our last point in this hybrid seed production, that is harvesting. So Optimum harvestage is when the seeds are fully mature, just before significant weather damage begins. So at this stage, seeds are easy to harvest and clean, resulting in minimum losses. So early harvesting will make the threshing and combining difficult. So it leads to high losses during cleaning and processing. Late harvesting, it will increase the risk of weather damage and it will cause the seed shattering and lodging. Again, ultimately it will lead to yield loss. Right? So this is complete about hybrid seed production principles and what are the operations we need to follow. I hope you got the valuable information from this session. So thank you very much. Thank you.